So today we're going to talk to you a little bit about some of the data analytic methods that Reed mentioned. This webinar is sort of a part 2 of the webinar we gave last fall. I believe it was in early November. So that's why we chose this title, Progress with iModulons and Comparative Pangenomics. So we're going to update you on a couple of these analysis methods. Most of the heavy lifting today will be done by Anand and Jason, and they'll take over after a brief introduction from me. So I may need a pointer, so there we go. So Anand will talk about our analysis of transcriptomes using ICA, and Jason will talk about new dimensions, new vistas in pangenomics. So the two data types that we are going to be studying are shown here. And I find this graph to the left always kind of informative. You're all familiar with this incredible drop in sequencing costs in the late 2000s, and by 2010, you can see that here, the price had gotten so low that it spurred a lot of sequencing work, and throughout the entire decade, we've experienced exponential growth in the availability of whole genome sequences for bacteria. And we have now downloaded all these data types, processed the data to sort out the sequences that are of suitable quality to do the analysis of the sort that we're doing, and have basically tried to deplete all the publicly available data. On the right, we have the same graph for bacterial transcriptomes. And you notice this takes off in 2013, not 2010 as over here. And that was primarily due to the fact that isolation of messenger RNA in bacteria is more difficult than in eukaryotes. No poly A tail to use here. But nevertheless, when that was in place, we also experienced an exponential growth in the availability of this data type. And interestingly enough, these estimated doubling times for the two of them are about the same. And as for the sequences, as Anand will describe, we basically depleted all the available, publicly available data at the moment for bacterial transcriptomes that are suitable for the sort of analysis that we do. So I hasten to add that in 1.7 years from now, there'll be twice as much data, so all of this has to be repeated. So it was our graphical summary or abstract of the talk from last November, where we went over 3 of these data analytic methods. So one of them shown here on the far right was to use binary phenotypes, resistant and sensitive phenotypes of pathogens, build metabolic models for them, use support vector machines to separate these the best we can based on variables that related to the activity state of certain alleles. So the alleles encode enzymes in this case, so the variables here are actually activities. And so the separating plane in this case is not just a plane in a high-dimensional space, but it actually represents explicitly a metabolic process whose activity separates the resistant data types from the resistant phenotypes from the sensitive phenotypes. And we tentatively called this approach FBA-GWAS or ML-GWAS. So we're not going to talk much about this today, maybe just touch on it a little bit. We've been doing pangenome analysis, quantitative pangenome analysis since a paper we put out in PNAS in 2013, cited here. And this continues to amaze us. So with every new scale in available genomes, we learn something new. And we'll talk a little bit about that. And then Anand was the pioneer of developing ICA methods to isolate signals in transcriptomes that are mixed signals generated by transcriptional regulators and isolate their individual activities. And we like to draw this analogy to an orchestra playing with a bunch of microphones in the room. and ICA is able to isolate the sound from every instrument because they have slightly different volumes at different locations in the room. So that is something we talked about last November, and as was mentioned in the introduction, the recording is available, the recording of that talk. So what are we going to do today? We're going to talk about a few things. So pangenomes, a lot of pangenome analytics were covered in the webinar in November, but today Jason is going to expand on that, and we now deal with something that we call the allelome. So the pangenome looks at differential gene counts in different strains that have been sequenced. The allelomes go a step deeper than that in terms of nucleotide resolution, where we can classify a particular genome in terms of the actual alleles that it carries of the genes that are found in it. So Jason has taken a broad view of these two layers here, and it's even broader than what's shown over here, and will be the second speaker after me and go through that material. Anand is going to give us an update on the iModulon work, so I won't say much about that, since he had described that in detail in November, how this actually works. But he has driven something called the Modulome project, whose name is derived from the graph I showed before, because we are basically getting a comprehensive set of iModulons now calculated for all the species indicated in the slide earlier. Finally, I wanted to point out that we pioneered a new way of representing all the bits of information that can be associated with the genome, something we call the bitome. And we're not going to talk much about that today, but there was a paper on that that came out in NAR in late 2020. And basically this formalism speaks to the formulation of a binary matrix filled with zeros and ones, whose columns are the individual nucleotide positions across an entire genome. So for the E. coli strain where this publication focused on, there were 4.6 million columns in this matrix. So then we could associate different features with the sequence. And it's actually astonishing when you start looking at it, how many different features can be associated with a nucleotide position in the genome. And so this is basically a presence-absence call matrix of whether that feature is found in that nucleotide position. And we have about 1,500 rows in this matrix today. So this is a comprehensive binary version of all the pieces of information that we can attach to a particular sequence. And we are continuing to look at this for new organisms and now beginning to look at their bitomes, and I'll just leave it at that. But I just thought I should introduce to you that this is a new organism that would be the finest resolution for describing information, all the information that we have for a given sequence. So that's the setup I have, and I would like to hand it over to Anand now, that's going to talk about the modulome and other exciting developments with iModulons.
All right. So just to double check, you can hear me, right? And see the screen? Great.
Yes.
So I'll start by talking about what an iModulon is. So we talked about this in the November webinar, but I'll give a quick review of how we calculate this and what we use this for. So in our 2019 paper, we had an expression compendium for E. coli that had 278 different expression profiles across the entire genome. We then applied this algorithm called independent component analysis that pulls out independent signals from a data set. And we looked at these individual signals, and these tells us what genes are co-expressed. So we called these groups of genes in one of these signals an iModulon, as these are independently modulated groups of genes. This name is kind of borrowed from Regulon with the ON ending, and it represents an expression signal. For example, a transcription factor that's sending a signal across the genome to upregulate certain genes. we found that a lot of these iModulons, or one important part of an iModulon is the gene weights. So you can see that for a specific signal, you have a value for all of the different genes, but most of these values are near zero. So only the significant values are kept in an iModulon. And what we found is these genes that are significant in each iModulon tend to overlap very, very strongly with characterized regulators. So ICA simultaneously calculates an activity matrix, and this tells us when are these genes expressed. So if we look at this expression or this activity spectrum across all the different conditions, each bar represents the activity of these genes. So you can see that some conditions have very low activity near the baseline, and others have very high activity, which means that these genes are highly expressed. So here's an example of one of these iModulons. This is the pure R iModulon in E. coli. And you can see there's about 13 genes up here that are significant and 2 genes that have negative weights. These 13 genes, we can map them onto the purine biosynthetic pathway. And we can also see that one of these genes with negative weights has to do with purine degradation, adenosine deaminase. So whenever these genes are upregulated, this gene and these 2 genes are actually downregulated, and vice versa. So we can look at the activity of this iModulon across a couple of different conditions. So minimal media is our baseline condition. So everything is relative to that. When we add adenine to the media, we see that pure R represses these genes, and it downregulates these genes and upregulates these genes. When we knock out pure R, it's de-repressed. So these genes increase in expression. And rich media has a very similar effect as adenine. So there is this one gene down here, YDHC, that's uncharacterized. So we've looked at the genomic vicinity, and it's actually right downstream of a putative transcription factor with no known function, YDHB. And we recently characterized this as a novel adenosine transporter in E. coli that had not been discovered before. That's regulated by YDHB, which is activated by adenosine itself. So this is one example of how iModulons can help us characterize new genes and new transporters. So another nice property of iModulons is if we apply ICA to 2 different data sets, we end up getting a lot of the same iModulons. So here's an example of 2 iModulons that were calculated from 2 different data sets where the genes are almost identical. And when we looked at these genes, we didn't find any common regulator that regulated all of these genes. But a lot of these genes had to do with periplasmic chaperones, membrane-bound proteins, and a lot of uncharacterized genes. And when we looked at the activities of these iModulons in these different data sets, we saw that when it was treated with antimicrobials, specifically membrane-associated antimicrobials such as polymyxin B, colicin, and combination therapy of cefsaludin and mesalinum, these were the conditions that had the highest activity of these genes. So this is showing us how iModulons can help us discover novel regulons that could lead to understanding of antibiotic resistance and just the response to antibiotics for different organisms. So like I described with those 2 previous vignettes, every iModulon has its own story. So this is kind of a tree map of all the 92 iModulons that we got from E. coli. And the boxes are relative to the size of the iModulons. And so an important distinction we want to make is between data-driven iModulons and the regulons that we get from literature. So the regulons from literature are based off of biochemical assays. And sometimes we may miss certain conditions when we're getting regulons from the literature. However, the data-driven iModulons are purely pulled from the data. And this is telling us exactly what genes are co-regulated together at all times. And so we used iModulons in a couple of different publications in the past year. For example, we used them to understand the response to heterologous gene expression, to look at a trade-off between growth rate and oxidative stress in aerobic respiration, to characterize mutations in OxyR, which is an oxidative stress regulator, and to regulate or to elucidate a few of the 2-component systems in E. coli. So we also computed iModulons for Staph aureus and Bacillus subtilis. So Sagat and Kevin led those projects respectively. And Kevin continued to actually create iModulonDB.org, which you can go to and explore all three of these iModulons. You can get interactive plots for each of the activities and the gene weights. You can search for genes of interest, and you can also download the full data sets and look at it yourself. So this was published in the 2020 Bioinformatics database issue. And this database is actually was designed to be scalable to new data and organisms. So because we have all of this tools in place, we decided to look at all of the available data from microbial RNA-seq data in the NCBI Sequence Read Archive database. So this is a graphical description of that. Here we see this is about 1,000 data sets. So there's a lot of data sets across a lot of different classes of bacteria and many species. So one thing that we noticed was there's no annotated strains for a lot of species and a lot of the data sets in SRA. So we had to do a lot of manual curation. So all of these hatched lines here represent data sets that were deposited that didn't tell us which strain they were from. And this data is from August 20th of 2020. So it could be significantly larger now. But what we did is we selected 13 bacteria and 1 archaea to download all the data from NCBI and process all of this data. So we've boxed right here all of the organisms that are probably relevant to the UO1 section, some infectious diseases. And for each of these organisms, we selected a model strain for the organism, processed the data, did some quality control, manually curated the metadata by going back into the literature and getting information such as carbon sources, strain identifiers, any stress or antibiotics that were used. And then we ran ICA on this to compute iModulons for each of these organisms. So this was a pretty hefty this was a pretty tedious effort, but we were able to compute iModulons for all of these strains, and we were able to start characterizing some of these iModulons. So what it means by a characterized iModulon is that we were able to associate either a biological function or a transcriptional regulator to that set of genes that was in the iModulon. So in order to facilitate this characterization of iModulons, we built PiModulon, which is a Python package for analyzing iModulons. So this was designed for a minimal coding background. So if you're able to pick up a Jupyter notebook and run it, then you should be able to use the PiModulon functions. And so the features of this are one-line gene set enrichment analysis, for example, TRN enrichments, gene ontology, or KEGG pathways, very quick publication quality visualizations, motif searching, and automated page creation for iModulonDB. So for all of these organisms that we've talked about before, the end goal is to put these onto the interactive iModulonDB page so that anyone can look at and see what genes are regulated by what transcription factors. And so here's a taxonomic tree or phylogenetic tree of all the organisms that are in the modulome. And we have a different person working on each of these organisms. So we have people working from master's students to postdocs in our lab. And we also have a visiting student from India and a collaborator in Korea that are also working on some of these organisms. So now I'm going to go into a few of the specific insights that we've gained for some of the infectious diseases using iModulons. So Pseudomonas aeruginosa has quite a few ways of dealing with antibiotics. For example, it has 3 sets of efflux pumps, this MEX-CD, MEX-HI, and MEX-EF. And it also has beta-lactamases. And these are all regulated by different transcription factors. And some of this is already known. So for example, MEX-CD is regulated by NFXB, MEX-HI is regulated by the SOXR system, and MEX-EF is regulated by MEX-T. When we performed ICA on the Pseudomonas aeruginosa data, we actually found that there were 2 regulons, or two iModulons that were associated with MEX-T, and both of them contained this MEX-EF transporter. So we now think that there's actually multiple regulators that, that affect this MEX-EF transporter. And we can also get the specific activities of each of these iModulons under each of these conditions. So for example, when we treat any of these Pseudomonas aeruginosa with any of these antibiotics that are listed here, we see that the MEX-T1 iModulon specifically gets upregulated. However, when we treat it with meropenem and ceftazidime, we see that specifically only the AMP-R iModulon is activated. So we also see antibiotic responses in Acetobacter baumannii. So this is showing an iModulon that is specifically activated by ciprofloxacin. It has 68 genes, including the TRAF system, which contains an antibiotic resistance island, which is shown here. It also has OMP-W and outer membrane protein, some iron-related genes, and subtilicin-like peptidases. And we can see that this group of genes is specifically activated when we treat the cells with ciprofloxacin. So this is showing us a set of genes that could potentially be used to understand antibiotic resistance and treatment and response in A. baumannii. Another interesting thing we found is that the fur iModulon is specifically affected by ciprofloxacin. So this is another visualization of the fur iModulon. This is just showing the genomic location, and all of the dots are a different gene. So genes above this threshold are in the fur iModulon, and that includes the BAU, BAS genes, siderophore biosynthesis, etc. And this iModulon has a pretty high overlap with the known regulon, but it does contain quite a few genes that are not known to be regulated by fur. So this is a potential for discovering more genes that are regulated by the ferric uptake regulator, or fur. So when we treat this, when we found a data set that treated the cells again with ciprofloxacin, and we see that the fur activity goes very far down. So all of these genes are repressed when we treat ciprofloxacin, when we treat A. baumannii with cipro. And interestingly enough, we actually did another study in this lab where we treated E. coli with cipro under different media conditions, M9 RPMI with LB and CA-MHB. And we found that under these different media conditions, the fur iModulons had different effects when we treated it with ciprofloxacin. So there's clearly a link between cipro and fur and iron concentration and iron uptake in these different bacteria that needs to be discovered. So one of the great things about getting iModulons for so many different data sets across all this phylogenetic tree is we can actually start to compare these iModulons and see how regulation quantitatively changes between these organisms. And so in this chart over here, each individual point here represents an organism, and they're connected if the iModulon in this organism is similar to the iModulon in another organism. So this is, for example, the translation iModulon in one organism, the translation iModulon in another organism. So this iModulon contains all of the ribosomal subunits, and we can see that these are very highly conserved across all of the organisms, except for, of course, the archaea. And we also see that chaperones are also highly conserved across all of these organisms. So this is the GRO-EL subunits. We see sulfur assimilation, iron acquisition, a lot of these very basal systems or functions of the cell are conserved across different organisms, and they're conserved in their regulatory roles. And so we can use this iModulon orthology to discover new regulons in bacteria. So for example, there was this one A. baumannii iModulon that overlaps very highly with the cis-B iModulon and CBL iModulons in E. coli. So cis-B is a sulfur uptake regulator and CBL is another sulfur uptake regulator, both of them in E. coli. And actually just 2 months ago, this paper came out showing that GIG-C, which is a ortholog of CBL, is required for cysteine metabolism and virulence and probably regulates all of these genes here in this iModulon. And so similarly, we see that this RPOH iModulon in E. coli shares quite a few genes with the Acetobacter baumannii iModulon. And the RPOH is the protein folding chaperone iModulon regulator. And over here, we see that there's a lot of genes here that are part of post-transcriptional or post-translational modification, protein turnover, and chaperones in this A. baumannii iModulon. So we think that all of these genes are also regulated by the ortholog of RPOH in A. baumannii. There's also one gene down here that's uncharacterized and fairly short, And this could potentially be an anti-sigma factor since RPOH is a sigma factor. And this gene has the opposite expression trend as the rest of the genes in the RPOH predicted regulon. So in conclusion, we're now computing iModulons for 14 different prokaryotes across the phylogenetic tree. And we're building this modulome, which is going to be a resource for anyone to look at for gene expression and regulation for all high quality publicly available data sets. And so our goal is to put this onto iModulon DB for anyone to interact with and look at the data. So we also showed that iModulons can lead to gene function discovery. And we've specifically looked at how antibiotics can induce expression of specific iModulons. And finally, we show that by building these iModulons for so many different organisms, we can use orthology to lead to regulon discovery. So with that, I'll hand it over to Jason, who's going to talk about comparative pangenomics.
So I'll talk about some techniques that we have been developing in the domain of comparative pangenomics. So briefly, this concept of the bacterial pangenome was developed to address this observation that many bacterial species have a very high level of intraspecies genetic diversity. For example, 2 strains of E. coli can share less than half of their genes, and consequently reference genome-based analyses are unable to capture the full extent of genetic diversity in any one species. So to work around this pangenome concept was developed, and most simply put, it is just the set of all genes observed in a set of genomes, usually of a given species. And the pangenome allows for analysis of a variety of different measures of genetic diversity for a given species that usually fall into these 3 categories. First, distribution, how many strains or subtypes have a given gene. Second is function, which can be a generic functional annotation such as Go terms or COG functional categories, or more specific types of functions such as metabolic functions, antimicrobial resistance, or regulatory functions. And finally, sequence variability, which can be the analysis of individual sequence variants that we refer to as the allelome, or individual mutations in those variants relative to some reference. And so by analyzing one or more of these dimensions in the pangenome, we have a variety of different methods for characterizing genetic diversity in a pangenome for a given species. And just along the spectrum of specificity from the most global to specific, we have analysis methods that begin with just the overall pangenome shape, such as its size and openness, and also trying to categorize genes broadly based on their frequency, most commonly trying to identify the core genome of a given species, or the set of genes that are present in nearly every single strain of that species. Then moving on into gene level analysis, we can start bringing in functional annotations of individual genes to see how specific functions or functional groups are distributed throughout the pangenome. And then finally, at the most specific allele or sequence variant level, we can begin analyzing individual variations in specific variants of a given gene observed in the pangenome and how those are distributed through the pangenome. And finally, interpret the individual variations between these variants from a structural standpoint by comparing the position of mutations to structural or domain annotations. So I'll be talking about some methods along this spectrum of scale, that have been generalizable enough to apply to a variety of different organisms and allow for comparisons between their pangenomes. So to begin, we had just over 12,000 genomes spanning 12 different organisms from the Patrick database. And these consist of the 7 escape bee pathogens, as well as 5 other World Health Organization priority pathogens, selected based on their infectious potential, as well as tendency to acquire antimicrobial resistance. So on the smallest end, we have the Enterobacter cloacae dataset at just over 100 genomes. And on the largest end, we have the Streptococcus pneumoniae dataset at over 3,000 genomes. And overall, these datasets appear to be pretty well distributed from a subtype perspective. Looking at the MLST subtype distributions, most of these datasets have dozens, if not hundreds, of different MLST subtypes represented, with the exception of this one case, second from the top, Enterococcus faecium, which is dominated primarily by this one subtype at 70% of the dataset. But we do adjust some of our methods coming up to account for this potential subtype bias. So in order to construct pangenomes from these genome collections, we use this three-step process. First, we identify all the open reading frames in all the genome assemblies of a given organism. We then cluster those ORFs by their protein sequence. And then finally, we can treat each of those sequence clusters as a putative gene, and each member of those clusters as individual sequence variants or alleles of that gene. Finally, we can then model the genetic variation or represent the genetic variation of each genome as a binary vector based on the presence or absence of each gene and allele identified by this process. And what this achieves is that it is able to consolidate the total variation in the coding genetic content of a collection of genomes into a neat binary matrix that is amenable to mathematical analyses without having ever relied on a reference genome. And so beginning the analysis at kind of the most broadest level of the overall pangenome shape, we looked into these properties of pangenome size and openness. And intuitively, openness is a description of the tendency for new genomes of a given species to introduce new or never before seen genes to that organism's pangenome. And so this property is typically most commonly quantified using Heap's law, which models the size of the pangenome as a power function of the number of genomes sequenced. And between the two fitted parameters k and lambda, this lambda parameter is one measure of this openness property, wherein a smaller lambda corresponds to a smaller growing pangenome and hence a generally more conserved organism, whereas a larger lambda corresponds to a faster growing pangenome and a generally more genetically diverse organism. And so typically this Heap's law is fit by randomly shuffling the order of your genomes and fitting the resulting empirical P of n curves and reporting the mean or median of the fitted parameters you get from these genome shufflings. However, given that we had this MLST subtype bias in some of our data sets, we decided to control for this by instead of shuffling all the genomes for each of these iterations, we would sample just one genome per MLST subtype. And to evaluate this MLST controlled Heap's law against the standard all genome Heap's law, we would fit the Heap's law to the first half of genomes in each iteration and see how well it predicts the pangenome size in the second half of genomes by either method. And ultimately, we find that this MLST controlled Heap's law tends to predict the pangenome size more accurately than using all of the genomes in a majority of these cases. Specifically, in 9 out of the 12 organisms we looked at, the MLST controlled approach more accurately predicted pangenome size. And of the three cases where it underperformed the all genome approach, one case was essentially a tie in performance, and the other 2 cases corresponded to organisms that had a good initial distribution of genomes from a subtype perspective. So now looking at the actual openness values that we got from this MLST controlled Heap's law, we have on the left the organisms sorted by the openness estimated using Heap's law and color-coded by their phylogenetic class. And we find that the openness values we get from this approach fall into these 3 clusters that separate roughly by their phylogenetic class. Most strikingly, this cluster of the most open organisms correspond to the 6 gamma proteobacteria in our data set. And these 2 smaller clusters for less open organisms are somewhat distributed with regards to the 3 bacilli species that we looked into, but also do a good job of separating out the other proteobacteria classes in our data set. So overall, this result seems to suggest that this pangenome openness as a species level property follows roughly the phylogenetic classification of that species. And so now moving on to more gene level analysis, we first looked into these gene frequency distributions, which essentially asks how many genes are observed in what fraction of strains for each organism. And we find that all of these gene frequency distributions pretty much follow this 2-peak shape. You have a peak of unique genes or a large number of genes that are observed in very few strains. You also have this core gene peak where a large number of genes are observed in nearly all strains for each organism. And so just another way this manifests visually is in the cumulative distribution taking on this rotated S-shaped curve. And so these two plots are just the example for the Campylobacter coli case, but essentially shows for all 12 of the organisms we looked at in these smaller plots here, where now the y-axis is on a log scale, and it shows pretty clearly this asymmetric two-peak shape that the gene frequency distribution takes on for all of these organisms. And so now, further exploring the shape of these two peaks, we find that on a log-log plot that these peaks pretty much appear to be linear and suggest that they follow a power function. And consequently, we decided to model these gene frequency distributions as just a sum of two power functions. And when we did this, we find that that functional form gives a very good approximation of the gene frequency distribution for all of the organisms, with an R-squared exceeding 99% for all but one organism, and even in that worst case, we had an R-squared of 96%. And so this functional form is able to represent the gene frequency distribution of these organisms quite accurately with just 4 parameters. And one practical use that we've used this functional form for is to segment the pangenome into common frequency categories, such as core, accessory, and unique genomes of each organism, based on the inflection point of this functional form. And one advantage this has to the more typical approach of setting fixed cutoffs, such as saying core genomes are genes in over 95% of strains, is that this approach takes into account the specific shape of the gene frequency distribution of the organism you're looking at and appreciates the asymmetry in these distributions when calling the pangenome frequency segments. And so when we use this approach to construct the core and accessory genomes of each of our organisms, we first find that the core and accessory genomes are similar in size for most organisms. The accessory genome tends to be within twice the size of the corresponding core genome and often much closer than that. And when we bring in individual gene functional annotations, we find that there are consistent functional enrichments in the core and accessory genomes of these organisms. For instance, in most of these core genomes, in most of these core genomes, we find significant enrichment for genes, for ribosomal genes, protein processing genes, and a variety of different metabolic genes, with a statistically significant enrichment observed in 9 to all 12 of the 12 organisms that we looked at. And similarly, we find in the accessory genomes consistent enrichment in more specialized functions. For instance, 9 out of 12 accessory genomes had significant enrichment in genes involved in trafficking and secretion, and 7 of the 12 accessory genomes had significant enrichment in genes involved in defense mechanisms, which can include functions such as those involved in antimicrobial resistance or the destruction of foreign DNA and RNA. And so overall, these results seem to suggest that there's some consistent structure by which pangenomes distribute genetic functions in multiple species at the gene level. And so finally, moving into the most fine-grained analysis of looking at individual alleles or sequence variants, we use this approach that we call the entropy of the allele distribution. This is based on the Shannon entropy H, which is a common diversity index used for characterizing species diversity in ecological communities or microbiomes. But in our case, we've applied it to the allele count distributions, which aims to summarize the extent of sequence diversity of a given gene based on both the number of unique alleles that it has, as well as their relative frequencies. And so this approach basically involves you count the frequency of each coding variant observed for each gene to get the allele count distribution for that gene. You then plug in that distribution to the entropy formula to get this summary statistic for how sequence diverse each gene is. And then we can sort the genes by this allelic entropy measure to identify the most conserved or most diverse genes. In this case, we look at the bottom 5% as being the most sequence conserved genes and the top 5% as being the most sequence diverse genes. And so this approach isn't limited to just the coding region. We also extended it to look at the sequence variability in the immediate flanking intergenic regions. Specifically, we did similar variant counts for the 300 nucleotides immediately upstream and downstream of the gene as observed in each pangenome. And by plugging in these counts into the same entropy measure, we get 3 different entropy measures for characterizing the level of sequence diversity, one for its coding diversity, 5' intergenic diversity, and 3' intergenic diversity, and ultimately yielding 3 metrics by which we can rank genes by their sequence diversity. And so again, we look for functional enrichment in these categories, functional enrichments within the top 5% of most conserved genes and top 5% of most sequence diverse genes based on either the coding region, 5' intergenic region, or 3' intergenic region. And we find that ribosomal genes are consistently conserved at the sequence level, not only in their coding regions, but also based on their flanking intergenic regions, with significant enrichment in 7 out of 12 of the organisms. However, we don't find any other functional group to demonstrate this sort of consistent conservation or consistent level of high variation across multiple organisms from either a coding or intergenic standpoint. And this result seems to suggest that while we saw this consistent structure in how functions are distributed at the gene level, at the allele level, there's quite a bit more variability in the functions of genes that have the highest or least level of sequence diversity. And so finally, we wanted to look into the actual positions of the sequence diversity across different organisms. And we focused on this case study of the ALK-A gene, which is a DNA damage repair protein specifically against alkylation damage. And this gene was selected based on the observation that it was classified as a core gene in 5 of our organisms, but among the top 5% most sequence diverse genes in 4 out of 5 of those organisms. And to analyze the position of sequence diversity, we employed entropy once again in a slightly different manner. For each organism, we would take all of the alleles of ALK-A, do a multiple sequence alignment, and then compute the entropy at each position to identify potential mutation hotspots or notably conserved regions in ALK-A for that organism, and then compare those against structural domain annotations. And so what this analysis yielded is first on these top 5 plots, we get the entropy along the length of the gene, where peaks correspond to probable mutation hotspots and troughs correspond to more conserved regions. And in the bottom 5 plots, we have the domain annotations along the length of the gene, which was our means to unify the location of these mutations and conserved regions across different organisms. And so by superimposing all these plots together, we first see that the location of mutation hotspots tend to vary quite a bit from between different organisms. However, conserved regions, there were 2 areas that were notably conserved across all 5 manifestations of this gene in these organisms. For instance, towards the center of the ALK gene, we have this region here that is conserved across pretty much all 5 organisms, and is located between the ALK N-terminal domain and this helix-hairpin helix GPD domain responsible for nonspecific DNA binding. Additionally, we get a similar scenario here of also universal conservation of ALK at this region, which corresponds exactly to this alkyl-based DNA glycosides domain, which contains both the binding pocket and active site of the ALK enzyme. And so with this ALK enzyme case study, we find that the location of mutation hotspots can vary quite a bit between different organisms for the same gene, whereas conserved regions can be conserved quite universally for the same gene across multiple organisms. And so with these analysis methods, we are able to compare pangenomes of 12 different organisms at a variety of scales. At the broadest level of pangenome, we find that this Heap's law approach works better at predicting pangenome size if you control for subtype. And the openness values you get from Heap's law suggests that the openness of a given species follows its phylogenetic classification. At the gene level, we find that the gene frequency distributions can be well approximated with this double power functional form, and that certain functional groups are consistently enriched in the core and accessory genomes of various bacteria, suggesting that there might be some consistent structure by which functions are distributed in pangenomes at the gene level. And finally, at the most specific allele level, this allelic entropy approach revealed that the most conserved genes at both the coding and intergenic regions are predominantly the ribosomal genes of a given species, whereas the sources of high sequence diversity are quite functionally diverse between different organisms. And finally, with the ALK-A case study, we find that the location of mutation hotspots can vary significantly between different organisms for the same gene, whereas conserved domains may also be consistently conserved across different organisms. So with that, I will hand it off to Dr. Palsson to summarize.
Based on these presentations, I think you see that there is a continued need to use knowledge types to enrich big data, the analysis of big data sets. And the big data sets we've been working with here for whole genome sequences of bacteria and transcriptome sequences, we reached the point where we can do global analysis of the data types currently available in databases. And to me, this is quite astonishing that we can be working with essentially all the available data when we deploy these methods.